So this is my mock presentation, and it should be around 2 minutes. So here we go. So my project focused on heath and fungi, and my project was titled "Using eDNA to compare Collina vulgaris, heather root, Bedonia, vascular lichen, and soil associated fungal communities in different habitats." So I'll start off by talking about fungi and their diversity. So fungi are a very diverse kingdom. All fungi are heterotroph, meaning they must obtain their nutrients from external source, and they usually form an association with a partner to get these nutrients, or they're decayed. So, for example, lichenizing fungi, they form associations with other photosynthetic organisms to form lichens. Vascular mycorrhizal fungi form associations with plants, and they have a mutualistic relationship. Parasitic fungi are parasites to many plants, animals, and decayers decay, decomposing matter. This is just an example of their diversity, and each of these have a different role within an ecosystem and provide different sort of ecosystem functions. And now fungi have a very important role within heathlands, hence why I decided to look at fungi within heathlands. So fungi are the main decomposer within heathland habitats, and they're very important for biogeochemical and nutrient cycling. Now, interestingly, heathland habitats do differ when it comes to their sort of plant composition and abiotic factors that they experience, and yet no study has ever looked to compare the main fungal communities within different heathland habitats. So my project looked at heathlands on Lundy. There are two types of heathlands found on Lundy. The first being lowland heathland. Lowland heathland generally has high levels of plant diversity, high levels of soil organic matter, but also contains heather, lichen, and generally not too harsh abiotic conditions. But if you compare that to the other heathland found on Lundy Island, which is tundra heathland, this is defined as an area that has barren rock, low levels of plant diversity, thin soil, but again also has lichen and heather. Now, the communities I decided to study were the dominant fungal communities in both of these heathland habitats, these being the fungi associated with Cladonia vascular lichen, fungi associated with Collina vulgaris roots, or inside the roots, and the fungi found in the soil. So a little bit about what else my presentation will cover. So I've talked about the fungi and about Lundy Island heathlands. I'll talk about how the samples were collected for each of these Collina vulgaris, for each of these samples, which I then compared between the two heathland habitats. So I compared lichen between the two heathland habitats, roots between the two heathland habitats, soil between the two heathland habitats. I looked at soil organic matter, as this has been shown to significantly impact fungal diversity, and I'll talk a little bit more about this later and how this might impact the diversity which we found and community composition. I'll talk about how I managed to calculate the community composition using eDNA methods, and I will compare the fungal communities between between the two heathlands. Okay, so the sampling sites, I selected 5 sampling sites for lowland heathland and 5 sampling sites from tundra heathland. All the tundra heathlands come from south of the 3/4 wall of Lundy Island, and all the lowland heathlands come from south 3/4 wall of Lundy Island, and all the tundra heathlands come from north, which is signified by this red line on this graph. Each sampling site was made up of a grid, and it's gridded like this. This is based on Natural England protocol, and there are 25 soil samples collected at each grid, 4 lichen and 4 roots. And the roots and lichen samples are collected on the corner of each sampling grid. So lichen samples were collected, they're 3 centimeter by 3 centimeter by 3 centimeter blocks, and they are bagged individually per sample location. Root samples were collected in soil blocks, and then the roots were taken out of these soil blocks and cleaned until 2 grams were collected from each sample location. And soil samples were collected using 18mm soil auger, and this was again done 25 times and all bagged per location. So I also analysed soil organic matter. So soil organic matter is dead or decomposing matter from plants and animals, and it normally makes up the top layer of soil. Soil organic matter impacts fungal diversity, and this has been shown, hence why I decided to look at soil organic matter. Soil organic matter has been suggested to differ between heathland habitats, so this might be interesting as it might be a driving factor in differences in fungal community composition, and soil organic matter was measured using hash rate analysis. So hash rate analysis is what you see here. This is a furnace. What we did is I took 500, it happens at 500 degrees Celsius. I took the soil, I put it into this furnace, it would blast at 500 degrees, and then the inorganic matter is weighed. And because we measured the weight of the original soil mass, we can figure out how much soil organic matter is in each sample. The results. I found, as expected, lowland heathland had higher levels of soil organic matter than tundra. And the significance of this will become clear in my results of the community composition. So eDNA analysis was used. eDNA analysis is looking at DNA released from organisms into the environment, and it's useful to detecting species within roots and soil and lichen, especially because these are non-fruiting species. So you can't just go out there and do a fruits body survey of like mushrooms, which you commonly see. You have to use eDNA. And use 3 different methods of extracting eDNA, which were the most appropriate methods for the samples. I amplified them using PCR and use the primers on the screen. And then I used Illumina sequencing to provide the metabarcoding data. So my results. So what these results show is the difference in the community composition between tundra and lowland heathlands for lichen. So in this map, you can see where the tundra samples came from and where the lowland samples came from. And you can see there's an overlap here. And this was seen in my NMDS, but confirmed using a permanent statistical analysis. And I found that there is an overlap between the community composition of lichen and the community composition between lichen in the tundra and in lowland heathland. And this suggests that there's a lot of similarities between within the community. And this is likely to be due to climate differences. So research has shown that the biggest impact on community composition for lichen is differences in climate. And because Lundy Island is quite small, it's only 4.5 kilometers long, there wouldn't be too much difference in climate, even with slightly different abiotic factors impacting both heathlands, hence why we found overlap. In contrast, the fungi found within the roots of Collina vulgaris did differ and also statistically differ, with no overlap seen in this NMDS between the tundra heathland and the lowland heathland. Now, it's likely that this is due to plant health. Research has shown that soil organic matter, which is found to be lower in the tundra heathland, can have a significant impact on plant health. And plant health drives the diversity and community composition within the roots. So this is why I believe there's a difference in community composition between tundra and lowland heathlands for root-associated fungi. And finally, a similar result for soil. I found that there was statistically a difference between the tundra and lowland heathlands community composition, as you can see, there's no overlap. And this is likely due to differences in soil organic matter. So the lowland heathland had a high level of soil organic matter, and a Shannon index found that they also had higher levels of diversity there. So soil organic matter correlated with diversity. And this is expected because with higher soil organic matter, there's more organic compounds, which would allow for a large diversity of fungi and therefore due to increased levels of different niches. So in summary, I found that the community composition of lichen-associated fungi overlapped between the tundra and lowland heathland, likely due to climate change, but future researchers focus on covering a larger span of land rather than just 4.5 kilometers. For root-associated fungi, I found that tundra heathland were significantly different to lowland heathland. Future researchers focus a bit more on plant health, maybe looking at not just soil organic matter, but either taking visual inspection of each plant's health, or maybe looking at chlorophyll levels. And finally, for soil organic matter, again, found significant difference between lowland and tundra heathland. And there are other ways of measuring soil health, maybe other than soil organic matter, such as soil density, and maybe this could be looked at. So why was this research important? Well, I found that different heathlands support different fungal communities for root and soil. I found that different fungal communities can result in different. It's important because different fungal communities can result in different ecological functions. So to maintain these different ecological functions and diverse fungal community, different heathland habitats should be maintained, and this should influence sort of habitat and conservation strategies. Thank you very much for listening, and I hope you enjoyed.